Hey guys, it's Mr. Dunbrook coming to you live or pre-recorded from my basement, depending on how you're watching this. I know that many of you don't have me as a teacher. Some of you have Mr. Summers. Some of you have, sorry, let me get this out of here. Some of you have Miss Ward or Mr. Hazer and all, but I'm going to be going through this PowerPoint for this unit for all of us, so I hope that's okay with everybody. Also, I hope you're staying safe and that you're taking care of each other. Very important. Before we even start this PowerPoint, a couple of quick things. Make sure you're also reading your textbook chapters. Those are posted on Google Classroom, or you have a physical copy of your book at home. Make sure you're staying up on your reading packets that are assigned to you through all your teachers, all your biology teachers in Google Classroom, and make sure you're submitting those assignments correctly and getting them in on time and staying on top of things. These assignments are going to include reading packets, vocabulary assignments, maybe summaries that you might write or type. So just make sure you're following directions in Google Classroom, please. Very important. Also, before we really kick into this whole business, let me get my face off the screen so you can focus on the good stuff here. Keep in mind that you guys just learned the chapter on genetics, so this is really kind of an extension of the genetics chapter. We want to know, what are genes made of? How do we pass this information on from one generation to the next, and how do they take that information and make their own traits with it? And so we're going to be looking at this big, long, fancy name, deoxyribonucleic acid, which is what DNA stands for. And DNA's cousin, or helper molecule, RNA, ribonucleic acid, that also has a lot to do with how we actually get our traits from our information that we get from our parents. Think of DNA as an instruction manual. If we know how to change the instructions, we could also fix mistakes. So we could cure genetic diseases. However, that's a double-edged sword. Some people may be interested in doing some things with DNA that could be not so nice. For instance, creating superhumans or something like that. So we have to be careful with what we do with this knowledge, because it's very powerful. This is the roots of life. This is understanding the very, very basic instructions of how to build life, which is pretty powerful stuff. So we're going to talk about some things as we go. We're going to first talk about how they discovered what DNA actually is and how it actually worked and how it was kind of organized, what we call the structure of DNA. Then we're going to talk about how DNA copies itself or replicates, because that's really important in order to pass your genes on to the next generation. Otherwise, everything would go extinct. And then finally, we're going to talk about how does this chemical instruction of DNA, how does it actually lead to a physical trait, such as, let's say, brown eyes? And the summary of that, don't worry about this yet, but the summary of that is, in other words, how do we go from DNA, this chemical, to trait? For instance, brown eyes. And it's going to involve a couple of intermediate or middle of the road processes, things that you probably don't know about just yet. So how do we go from DNA, from what the genes we got from our parents, the chemical code or instructions we got from our parents, to actually physically having a trait? And it gets a little bit complex, but it's going to basically involve DNA sending its message to its helper molecule, RNA, and RNA helping to build what we call a protein enzyme. You remember enzymes from chapter 2. They are proteins that control our chemical reactions. So we build a different protein to get a different chemical reaction, which gives us a different trait. For instance, if you had the DNA for brown eyes, your RNA would then help build a protein that gives you a chemical reaction, an enzyme that gives you chemical reaction that gives you blue pigment, which gives you blue eyes. However, if you have brown eyes, you would have a different chemical code on your DNA, which would then have your RNA build a different protein enzyme, which would give you a different chemical reaction, one that makes brown eyes, and you would get the brown-eyed trait. If I lost you on that, don't worry about it yet. Okay, I'm also going to go to this quick video. It's a kind of an overview to keep things in your head kind of straight. Remember, you're just coming off of this chapter from Gregor Mendel, where he was working in the 1600s, and he knew that heredity had these patterns and that we were getting dominant and recessive alleles, he called them, from mom and from dad, but he didn't know the actual chemical makeup of what those genes or alleles were. So that's what this unit's going to focus on. A quick video to kind of sum some of this up and put the big picture in place for you here. I may talk over this video a little bit.
Despite visible differences, the molecular level, humans are 99% the same. We possess 46 chromosomes, 23 from each parent. Each chromosome holds an assortment of genes, These genes create different proteins that control a specific structure or function within the human body. Genes are composed of DNA, a long, double-stranded molecule made up of 4 nucleotides or proteins: adenine, thymine, guanine, and cytosine.
Now, it's important to realize all living things, from bacteria to humans, have those same 4 chemicals, A, T, C, and G, in their genetic code. So how could we possibly be so different from one another? Well, it's all about the order that you put them in. That's where we get our diversity.
Bonds that form between nucleotide pairs, A always pairs with T and C with G, hold the intertwining strands together in the shape of a double helix. Recently, scientists completed the Human Genome Project, a massive undertaking that sequenced all 3 billion base pairs in the human genome. The result is a huge genetic blueprint consisting of approximately 100,000 genes. Now that all of our human DNA has been mapped, scientists will identify which sequences are actually genes and which of the 100,000 genes are involved in specific diseases like cancer, heart disease, and diabetes.
Okay. Keep in mind, those 4 letters, again, are common to every living thing, and we're all different. So how do we get these differences? It's really just about the order that those A's, T's, C's, and G's are in any given individual. We'll get more into that later, but you probably noticed in the video that the A's were always bonded to the T's and the C's were always bonded to the G's. In our class, we used to say apples grow on trees and cows eat grass, A to T, C to G. But again, we'll get into that later. But just keep in mind, this is the stuff of genes, deoxyribonucleic acid. Here we go. It all starts with a guy named Frederick Griffith. He was curious as to how disease really got people sick. He was studying a disease you guys are probably familiar with called pneumonia. Well, it turns out there's a couple different bacteria that are kind of pneumonia-like bacteria. We call them pneumococcus bacteria. But one gets you sick and one does not. And he wanted to know, well, what trait did the bacteria that got people sick have that was different than the one that did not get people sick? So he did some experiments. Of course, in biology, we always have to kill a mouse, right? So what he did is he isolated, or got by itself, 2 slightly different strains of the pneumococcus bacteria. One caused death, the other was pretty much harmless. And he did some monkeying around with this bacteria. What he did was he took some of the bad bacteria, injected mice, and they died. He then took some of the good bacteria, or the less harmful bacteria, and injected mice, and they lived. But then he went a step further. So all the mice that got the harmless bacteria lived, but at the time, nobody knew in this experiment how the bacteria actually made you sick. They thought maybe it was a poison that made you sick. But if it was a poison, you couldn't kill it. Keep that in mind. So Frederick Griffith, here's what he does. To test this, remember there was 2 different strains of this bacteria. One was harmful, one was harmless. To test this, he heated a solution of the disease-causing bacteria to kill them. So he killed the bad bacteria, and then he injected them into the mice. They didn't die. So by injecting the bad disease-causing bacteria into the mice after he had already killed the bacteria, those dead bacteria did not kill the mice. I hope you're with me so far, because we're going to go a step further now. So this suggested it wasn't a poison, because you couldn't really kill poison. Poison's not alive, right? So he's saying to himself, well, what is it that's getting people sick then? So next, Griffith did this. He mixed the 2 bacteria together. He mixed the killed bacteria, the bad ones, with a solution of harmless bacteria that were still alive. So now you have the bad bacteria that's dead mixed with the good or harmless bacteria that's still alive. He knew neither one of these by themselves would kill the mice. He knew that the harmless bacteria would not kill the mice. He knew that the dead, harmful bacteria would not kill the mice. But when he combined them and injected them into the mice, to his surprise, they all died. So this started him wondering, what the heck's going on? How did these mice die? What was the actual chemical that was giving these bad bacteria the power to kill, or the power to have the trait to kill? And he did an autopsy, which is kind of what you do when you look at something that's already died. You You cut it open and check it out. So he autopsied the lungs of the dead mice and found them riddled with this bad bacteria. Somehow, and this is important, somehow the killed bacteria passed their disease-causing ability to the harmless bacteria. He called this transformation. Think of it this way. Think of it as a soldier without a gun and a dead soldier with a gun. Neither one of those could kill you by themselves. A dead soldier with a gun, a living soldier without a gun. Neither one could kill you. But put them together and they could kill you, because the harmless soldier that was alive could grab the dead soldier's gun and then become deadly himself. In this case, though, the gun or the weapon was really DNA. So we'll get into that a little bit later. So I hope that you're understanding that experiment. It's a little bit complicated, but it's kind of neat too. So he hypothesized, Frederick Griffith, that something in the killed bacteria was transferred to the good bacteria. Again, think of the gun going from the harmless soldier that was alive, the harmful soldier that was dead to the harmless soldier that was alive. The harmless soldier took the harmful soldier's gun and became harmful himself, okay? So he also hypothesized that it contained a gene with information that could cause change the harmless bacteria into the disease-causing bacteria. What really happened here was the harmless bacteria really got the trait in order to be deadly from the dead, harmful bacteria. Again, the soldier that was alive stole the dead soldier's gun. Frederick Griffith wanted to know how this happened. Really, he stole his DNA, or his DNA was transferred. So along comes a guy named Oswald Avery in 1944, a little bit later on, right? He repeats Griffith's experiment, but he did it with a little more detail. He did this to find out which molecule was actually responsible for the transformation. In other words, he wanted to know what molecule was passed from that bad bacteria to that good bacteria that made the good bacteria deadly. Now, today we know it's DNA. Back then, they did not know yet, okay? So what he did is he mixed up a juice, or just, in a beaker, of the killed bacteria. Then he went a step further and he treated the juice with enzymes that destroyed all proteins, lipids, carbohydrates, even RNA. The only thing that was left was DNA. I hope you remember some of these molecules from chapter 2. Carbs, proteins, lipids, all that stuff, nucleic acids. So we know that those are kind of the 4 macromolecules of life: carbohydrates, lipids, proteins, and then nucleic acids like RNA and DNA. So Oswald Avery basically got rid of all the carbohydrates, all the lipids, and all the proteins, and the only thing left was DNA. So he's kind of testing one variable at a time here to see if DNA is responsible for the transformation. Okay, let me pause this. I got to reload. So Oswald Avery, again, really repeated Griffith's experiment, but a little bit more intense. He was now trying to narrow down what the actual chemical was that caused that transformation of the bad bacteria to the good bacteria. So he did something very similar. He's mixing up different concoctions and injecting mice, but he is now able to tell if the mice got DNA, or if they got carbohydrates, or if they got proteins, or if they got lipids. In other words, he's narrowing down the chemical that is the stuff of genetics, or the stuff of genes. He came to realize, after he isolated all these different chemicals and shot them into mice, basically the ones that got the bad DNA from the bad bacteria are the mice that died. And you can read about this experiment in your textbook. I won't take up too much of your time with it, but it is pretty neat the way he isolated different variables and tested one thing at a time. He ultimately concluded that the DNA must have been the transferring agent. The DNA must be the transferring agent that actually led to the bad bacteria passing the trait on to the good bacteria that made it deadly. Okay? All right. Then comes Alfred Hershey and Martha Chase. Again, just a couple of scientists trying to kind of determine or put their finger on what was going on with this business of DNA. And they did some work with viruses. And believe it or not, this is actually pretty timely with the whole coronavirus thing. They did some work with viruses that affect or infect bacteria. And stick with me here, because these bacteriophages are actually viruses. So a virus that infects a bacteria is called a bacteriophage. I know that sounds like it's a bacteria, but it's actually a virus. And viruses are, remember, they're not technically quite alive. They're very close to being alive, but they're not technically alive. So bacteriophages are viruses that contain either DNA or RNA. They contain the chemical instructions for life, but they by themselves are not alive because they can't reproduce on their own. They need other cells in order to reproduce. That's the same with the coronavirus. By itself, it's not a living thing. It needs to get into your cells in order to reproduce. So that's why technically scientists don't consider it alive, but viruses are very close to being alive, and they're very good at what they do. So the bacteriophage attaches itself to the surface of the bacteria, and it injects its DNA into the bacteria. This then takes over the bacteria's DNA. So this is kind of like putting software in your computer, and the computer is now running your software, and it's working for you, doing what the software tells it to do. So the virus gene directs the bacteria's genes to make millions more bacteriophages. Remember, these are viruses. That's what allows viruses to spread. Is a virus will stick its instructions inside of your cells, and those instructions say, make more viruses. So it kind of hijacks your cells. It's like a hijacker of a plane. It starts flying your plane and directing the show. Or if you don't like that analogy, it's kind of like software slipped into your computer, and now your computer is doing whatever the software says. Of course, that software is really DNA or RNA. It's genetic instructions. Okay, so when this process is completed, the bacteria cells burst open and they release more virus and it kills more bacteria, and the whole process continues. Again, this is very similar to what's happening in people with coronavirus. Kind of an offshoot here, we can actually use that. We can use viruses to deliver instructions or DNA into other organisms. That's what they do best. And here's an example of an experiment where they took a harmless virus and they put a gene from a lightning bug, a firefly, into the virus. And then the virus infected this mushroom and infected all of its cells. So now its cells glow in the dark, and it gave those cells of the mushroom the gene or the trait, the chemical instructions of DNA to have the ability to glow, just like the lightning bug. So again, take the lightning bug gene out of the lightning bug, put it into a virus, put the virus into the mushroom. The virus then copies itself and gives all the cells in the mushroom the ability, the instructions to glow like a lightning bug. Pretty crazy stuff. Again, gotta be careful with how we do some of this genetic stuff. It could be used for bad purposes. So Hershey and Chase figured out that they could determine which part of the virus, the protein coat or the DNA, entered humans. Again, they're trying to nail down what is the chemical responsible for causing traits, for genetic instructions. Remember, Oswald Avery and Griffith were kind of getting onto the track, but these guys are just kind of cleaning up the details and further proving what Oswald Avery and Frederick Griffith proved. Real quick lesson on viruses. This is not a coronavirus. This is a different type of virus. Remember, they're called bacteriophages. These viruses infect bacteria, but they're basically protein coats. They have a protein coating, and they have some DNA or RNA inside, some chemical instructions. What Hershey and Chase wanted to know was which part of this virus was the instructions that got into the other cells and took over those cells. In other words, what's the software? Is it the protein? Is it the DNA? What is it? Okay. And to do this, what they did was they took some radioactive chemicals. Radioactive, think of those as glowing in the dark. You can kind of see them and trace them, and they're very easy to follow. And he took radioactive sulfur. By the way, sulfur is only found in proteins. And then he took radioactive phosphorus. Phosphorus, P, is only found in DNA. So this is going to kind of be like a marker or a tag to follow these two chemicals. And by following these two chemicals, you can tell if you see glow-in-the-dark phosphorus, you know it came from DNA. If you see glow-in-the-dark sulfur, you know it came from protein. So they're going to follow these chemicals to see which one, the DNA or the proteins, got into the bacteria cell. Okay? And lo and behold, I'm sure you can kind of hypothesize what happened when he did this. Don't worry about all the details here. But when he did this and he shot these, or he put these viruses onto the the bacteria, he found that the bacteria ended up having all phosphorus inside of them, which meant, remember, phosphorus comes from DNA. Phosphorus comes from DNA, okay? He did not find any sulfur inside. So what he concluded was it must be the DNA that this virus is shooting into the bacteria, and that's what the instructions are. So now we're narrowing down what is the stuff of genes. What are the chemicals of genes? Okay? And scroll up a little bit. So Hershey and Chase, this is a big concept here, concluded that the genetic material, the stuff genes are made of, is DNA, not protein. Very, very important concept. Okay? All right. I'm actually going to stop right there for today's video, and make sure you check out the next video and do any summaries that your teachers are asking you to do. Everybody stay safe. All right? Have a great day, stay safe, and take care of each other. We'll get through this. We'll get through this, and hopefully we'll see each other again very soon. Okay. Have a great day, everybody.